Hello everyone, we will proceed with the second part for natural products from microorganisms. I'm Dr. Atira from Biotechnology Research Institute, University Malaysia Sabah. There are 5 sections and we have covered 2 of them in the previous lecture. Now let's move on to the other sections. We will discuss the potential of first, bacteria as the source of antimicrobial proteins. Second, microbes as the source of antifungals and microbe-derived anti-cancer agents. Introduction on microbial natural products. So, we have learned that a variety of microorganisms produce secondary metabolites, also known as natural products. Now let's discuss on natural product applications. Natural products originate as secondary metabolites from a variety of sources, including plants, animals, marine organisms, microorganisms, terrestrial vertebrates, and invertebrates. A variety of biochemicals ranging from alcohol to antibiotics and in processing of foods and feeds. Microorganisms have great potential as natural sources of drugs for the treatment and prevention of diseases like cancer. They are also potential sources of natural antioxidants, colors, immunosuppressants, enzyme inhibitors, vitamins, enzymes, and antibiotics. The discovery of penicillin from Penicillium notatum in 1928 by Alexander Fleming marked a significant shift from plants to microorganisms as the source of natural products. The early years of antibiotic research discovered streptomycin from Streptomyces gracias and chloramphenicol from Streptomyces venezuela. Lactococcus lactis are very well known for centuries in the fermentation of food, especially in cheese and yogurt, are also being used as cell factories for the production of important nutraceuticals. Lactobacillus acidophilus, a probiotic, triggered the production of a beneficial immune molecule which helps to regulate the immune system. Among common probiotic bacteria are the Lactobacillus, Bifidobacterium, Saccharomyces boulardii. Prebiotics are types of dietary fiber that induce growth and the activity of gut bacteria to produce nutrients for colon cells and lead to a healthier digestive system. Now let's look on how antibacterial agents can be used for fighting bacterial infections. Several bacteria produce a variety of antibacterial substances, namely hydrogen peroxide, fatty acids, organic acids, ethanol, antibiotics, and bacteriocins. Antimicrobial peptides or proteins produced by bacteria are categorized as bacteriocins. Bacteriocins are antibacterial peptides or proteins that either kill or inhibit the growth of closely related bacterial strains. Generally, bacteriocins are only toxic to bacteria closely related to the producing strain. Example, several pediococcus strains can also be used effectively in food system to control Listeria monocytogenes. The killing ability of bacteriocins is to maintain population and reduce the numbers of competitors in order to obtain more nutrients and living space in environment. Many bacteriocins are produced by food-grade lactic acid bacteria. They act as natural preservatives to control the growth of spoilage and pathogenic bacteria in food. Unlike most antibiotics, which are secondary metabolites, bacteriocins are ribosomally synthesized and sensitive to proteases, while generally harmless to the human body and surrounding environment. In this table, you will see some examples of microbial-derived antibiotics such as erythromycin, tetracycline, vancomycin, streptomycin, niacin A, and deuterin. Some functions as antibacterial that specifically kills bacteria cells, while others function as antimicrobial agents to prevent the spread of bacteria, fungi, parasites, and other viruses. Next, let's have a look at the development of antifungal drug. An antifungal agent is a drug that selectively eliminates fungal pathogens from a host with minimal toxicity to the host. However, only a limited number of antifungal agents are currently available for the treatment of life-threatening fungal infections. These are the examples of fungal infections commonly treated with antifungal We have the ringworm infection, athlete's foot infection, and fungal nail infection. This is an example of antifungal agent. Nystatin is a Pauline antifungal agent. It is one of the first effective Pauline antifungal agent obtained from Streptomyces norsei. It is effective against Aspergillus and Candida species. It is used to treat fungal infections in the gastrointestine, mouth, genital area, and on the skin. Now let's go to the development of anticancer. Microbes can be used to make anticancer agents. Microbial metabolites are among the most important of the cancer chemotherapeutic agents. There are many microbe-derived anticancer agents that have been evaluated through clinical trials. For instance, the polycation actinomycin was isolated from Streptomyces parvulus in 1940. It was the first antibiotic shown to have anticancer activity. These are examples of microorganism-derived anticancer agents, example of compounds that have been isolated from microbes for cancer treatment are the actinomycin, bleomycin, taunomycin, doxorubicin, epirubicin, idarubicin, mitomycin C, gildanomycin, rapamycin, and wortamanin. You can see that the list of compounds are isolated from different types of microorganisms. However, there are challenges for the production of natural compounds as drugs. Many natural compounds with potential as drug candidates occur in low concentration in nature, often making drug discovery and development burdensome and economically impractical. Therefore, scientists have come out with an alternative solution, which is to express biosynthetic genes from the original producers in microbial hosts. These are examples of host strains that are being used to produce natural products. Their first example is Escherichia coli, E. coli, a gram-negative bacteria which is being used as a host to produce recombinant human insulin. Next is Lactococcus lactis, a gram-positive bacteria that is used as a host to produce tetanus toxin fragment C. We also have Streptomyces species that is used as a host to produce antibiotics such as tetracycline and chloramphenicol. Bacillus species is used to produce alkaline protease, while Saccharomyces cerevisiae is used to produce human serum albumin. Natural products have provided significant value to the pharmaceutical industry, particularly in the therapeutic areas of infectious diseases and oncology. More than 70% of FDA-approved antibiotics are natural products, all of which were derived from microbes. Chemical diversity from microbial natural products lead to development of novel drugs such as antibiotics, anticancer, and antifungal, and along with other pharmacological activities. However, the real challenges are microbial production of natural products involve low production titers, difficulty in production isolation and structural identification, and production of inactive proteins. A wide variety of engineering strategies such as genome editing, ribosome engineering, precursor engineering, mutagenesis, and overexpression of structural genes are used along with the conventional recombinant DNA technologies to facilitate the efficient production of natural products and pharmaceuticals in microbial systems. The CRISPR-Cas is one of the highly prospective tools for genome editing for additional improvements and to increase production of microbial natural products. If you have any questions, you can always email me. Thank you.